So welcome everybody. We'll go ahead and get started. I know a few more will continue to join us. I'm Diane Alps from the Cabrillo Marine Aquarium, and joining me tonight, let's see, to this side is Louisa.
Hi everyone.
Louisa from the president of our Los Angeles chapter of ACSLA, and we are so excited tonight to have our next deeper dive. So the deeper dive originated from, it was one of those COVID products really, but the Cabrillo Marine Aquarium and the American Cetacean Society Los Angeles chapter have been partnering for over 50 years. Our primary partnership involved training naturalists, many of whom are with us tonight, to go out on the local boats, talk about whales. It was one of the first volunteer programs of its type here at the kind of the birth of whale watching as we know it today. So we're really proud to continue on that long tradition and continue that partnership. And once we went into the shutdown, we had to transition a few things, and one of our traditions was also having an ACSLA public lecture on the last Tuesday of every night at the Cabrillo Marine Aquarium. And so we embraced technology as it is and created deeper dive through this partnership to continue reaching out to everybody and bring and this really gave us the ability to bring speakers from around the world, and we have had some really great ones, including tonight's, and I'm really excited to introduce you all and have a chance to chat with Daniel tonight. In the meantime, let's go ahead and take a second. I'll talk for just a moment about our partners. Again, I'm with the Cabrillo Marine Aquarium, and we are a small but mighty aquarium in San Pedro, very, very hands-on, really focused on education and interactivity. It's the start of tide pool season really here in Southern California. So if you're in the area and want to join us on a tide pool walk, or even if not, head to your local tide pool. It's a great time as we get into the winter season. It's a really great time to take advantage of the low tides and see that everything that nature has to offer. So I hope you'll come to the aquarium one of these days and give us a visit. And I'm going to turn it over to our other partner with Louisa.
Hi everyone, welcome tonight. I'm president of ACSLA, and we're here. We're so grateful that we're able to partner up with Cabrillo. We've been doing this for such a long time, and it's one of our favorite partnerships. ACS just had a national conference in San Diego at the beginning of the month, and I am pleased to announce that ACSLA was able to give away 3 grants to students. We call them travel grants. Usually we give away 2 that are about $250 a piece. This year we were able to give away 3 $500 travel grants to students, and they happen to be this year undergraduate students with amazing posters, and hopefully come 2023 they will be one of our webinar attendees will be speaking, and we'll have more information about that at a later date. I wanted to say that the reason we were able to give away so much money this year is because we had 2 donors who at the conference, actually, one is anonymous, who asked to remain so. She gave away $500, and then Marcia DeVoe, who is also one of our local members, gave away $250. So thank you both to these people for helping. And we were able to bring 3 students to the conference with this money. One of them actually came from Minnesota. But anyway, I just wanted to say thank you to everyone. And it's one of my favorite things to do as president, is to give out money to the future scientists, whale savers, if you will. It makes me feel good. So anyway, thank you, Diane.
Congratulations to ACSLA for being able to give those travel grants. They're much needed. I know that students really appreciate that extra income. School is not cheap these days. And, I know a lot of scientists will talk about their early grants often coming from ACS. So I know it's very, very valued and held near and dear. And if I'm not wrong or mistaken, I think a couple of those poster winners were actually actually from Oregon State as well. So how fun that we're connecting that. Well, thank you very much, Louisa, and congratulations on being able to give those travel grants. So we are going to turn it over to our speaker now. So hey, Daniel, you want to join me? Hi.
Hello there.
Hello and welcome. How are you?
I'm very excited to be here tonight with all of you. Thank you so much for the invitation. I'm glad we made it work. We had been going back and forth about when the right time was, and finally the stars aligned. So thank you for inviting me.
Exactly. I'm so happy you could join us. And first, I'm going to say I love the posters behind you. So it's a really nice backdrop, as though it's planned. But yeah, very nice whale
It's
art
my
in
office.
the background. I love that. So I hope you heard all the shout outs for Oregon State. And as I mentioned, I think that a couple of those travel grant winners that ACSLA was able to award were students from Oregon State.
Indeed,
Cool.
they definitely benefit from travel grants and awards, and it's a wonderful thing to have that resource for the students. So thank you.
Absolutely. And if I'm not mistaken, I think, I mean, travel grant, obviously it's supposed to cover the travel and maybe hotel a little bit, but it probably is about the same cost of maybe a book. So hopefully that helps to offset some of their costs, since books these days. Or have they gone digital and I'm just out of touch?
Yeah, that's about right.
Yeah, exactly. Well, we're really excited to have you tonight. Thank you for joining us from Oregon State. And I'm really looking forward to your talk. It sounds like you've created something kind of new for us, which is kind of the trend with Deeper Dive. We love to kind of pull out some interesting different things. So what gave you your inspiration for the direction you're taking us tonight?
Yeah, well, I like your idea of Deeper Dive being a little bit along the lines of a fireside chat. Most of the talks I give are more technical and geared to a scientific audience. And it's kind of refreshing to do something a little bit different, maybe a little bit more personal. And in trying to choose a individual topic, it was actually really hard to just come up with something that I felt excited to talk about. So instead, I thought that I could give maybe a bit of a trajectory of my path that I have followed to where I am, and maybe through a series of vignettes, maybe hit some points about special moments or things that have provided the next jumping off point to the next opportunity. So that was kind of like my approach to how I went about to put this together. But yeah, we'll see a lot of old photos from treasure troves and revisiting some of my longer deeper time history here.
That's excellent. It's I know you've had quite a journey, and I'm excited to hear it. You've dove in quite deep into many different areas of research, and particularly whale research. What are you doing right now? Where are you, and what are you doing? I mean, we've talked about Oregon State, obviously, but what are you doing there?
So I'm in my office because this is where I have the best connection and computer screens and all of that. And I am in Newport, where it's raining in Newport, Oregon. I'm at the Hatfield Marine Science Center, which is the coastal marine facility for Oregon State University. And there's a campus of about 300 people here, about half and half local, federal, and state agencies, as well as Oregon State faculty and students here. So yeah, that's where I am.
And you're running the basically the Whale Telemetry Lab with the WHET Lab, I believe it's called.
Now it's been renamed the WHET Lab, or we are the successors. But yeah, carrying a long tradition. Bruce Mate started here at OSU over 40 years ago, and single-handedly built a program. And I'll talk a little bit more about that in my slides, and I'll show you some of what we've been up to lately. But yeah, I'm the lucky person that gets to continue his legacy and his tradition.
Congratulations on that.
Thank you.
Congratulations. We're really proud of you and excellent. Glad that you're there. So let's go ahead and have you load up and we'll turn it over to you now. And we want to remind all of our attendees, we will be having, as in our tradition for Deeper Dive, we will have lots of chances to talk, being questions from you to get from our speaker. So please do post those in the Q&A. It's a little bit easier to manage and moderate those questions if you'll post them in the Q&A. And we'll take several breaks throughout. So please do that. Daniel, I think you still have to swap again. It looks like
Let's do that. Thank
it
you.
went back to that. Yeah, there you go. All right, I'm going to turn it over to you now, Daniel. All of you that are attending, remember to post in the Q&A. I'm sure there's going to be lots of questions. All right.
All right. So coming up with the title for this talk was actually the hardest part because I couldn't quite put it all together in terms of these disparate topics that I'll be talking about. So I just came up with this idea of growing up as a marine mammal scientist in terms of the challenges and opportunities and even some of the responsibilities that each of those steps involved and inevitably the adventures that followed. So thanks once again for having me and quite excited to be with you tonight. And thank you for being here. As I was saying before, I am currently in Newport, Oregon. This is the Central Oregon coast. And Oregon State University has a marine campus on the coast that houses a lot of our research that is ocean oriented. I'm very excited to come to you from the Gladys Valley Marine Science Studies building, which is our newest building and is quite a state-of-the-art in terms of construction for coastal along the West Coast because of the threat of tsunami. And what you see here on the right is the building doubles as a vertical evacuation point. So this ramp leads to the the top over the third floor of the building where, although I hope to never get to see that, presumably we will be able to withstand even the strongest earthquake and tsunami and take refuge there on the top. So that was part of the design element of the building to have that. And as I mentioned, we moved into this building probably about a year and a half ago. And we're quite privileged and quite excited to be in this building. The other development is that, as I mentioned before, Bruce Mate, who was the first marine mammal scientist based here at Oregon State University over the course of 40 years, built a program that eventually was named the Marine Mammal Institute. Bruce retired about 3 years ago, and we are very, very fortunate to have Dr. Lisa Balance as our new director. Some of you may know her because previously to being here, she was the director of the Marine Mammal and Turtle Division at the Southwest Fisheries Science Center of NOAA in La Jolla. So with her at the helm, we have been really taking things forward and occupying the new building. My office is actually right here. This is where I'm coming to you tonight from the third floor, that corner office. And it's all quite exciting to be part of this. Right now we have 7, in addition to Lisa, we have 7 faculty members that serve as principal investigators, and a total of about 50 people that make the Marine Mammal Institute, of which about 30 are students, graduate students, and some undergraduates as well. But again, thank you for the donations and the contributions for the American Cetacean Society grants program that our students have access to, because they definitely make regular use of that. So we also talked about my lab, the Whale Habitat Ecology and Telemetry Laboratory. And our role is to develop tag technology, advance the ecology, and inform the conservation of the largest animals on Earth, obviously whales. And here are the current lab members. I have 2 PhD students, Michaela and Ladd, 1 master's student, Liam. Clarissa is a postdoc, and Thomas Follett is a faculty research assistant. To go a little bit more detail about the vision of the lab, we have a 3-pronged vision where tag technology development, which is the legacy, the item that we carry forward from Bruce Mate, to address important but unanswered scientific questions in whale ecology, and to address interactions with human activities and the effects of human-driven factors. And our mission is to ensure that that information that we generate reaches the natural resource managers, and that is presented in ways that can be most effective in terms of improving the conservation of whales. I probably will not be saying too much about these things because these are the kind of topics that I normally address during more technical presentations, and this is a little bit more of an anecdotal personal journey, but I'm happy to talk about any of these topics if there's questions. Here are some of the pictures that we took this year during our field season in the summer with some of the same individuals that I identified in the previous slide. It was quite busy, quite exciting here off the Oregon coast, working with humpback whales, gray whales, blue whales, and even a fish, a mola mola, that came to our boat to say hello. Given that this is American Cetacean Society, I thought I'd just take a little bit of a sidebar to mention that the design of the logo for the lab was actually in collaboration with Uko Gorder, who is your national president, and a wonderful person. I got in touch with him, and knowing that his abilities, his skills, his profession as a natural history illustrator of great renown, I came to him with some ideas and asked him if he would help me design a logo for the lab. And I thought I'd share with you some of that process. So I just had this idea that I wanted to capture some of the ecology, some of the interaction of the animals in their environment. So I started with these images that I found on the web that I found particularly relevant or appealing or striking. And just a few more things, like in terms of elements of design, what the logo should have. So these are my rudimentary sketches on my iPad, and I basically provided this to Uko, and he came back with this. So yeah, as I mentioned, it was just a little side note to give you that that background story of how the lab logo came to be. So now let's really dive deeper, or start to dive into this journey that I mentioned earlier. So I'm from Colombia originally. I was born in Bogota, which is the capital. It's a landlocked city up in the Andes Mountains. But I grew up obsessed with watching TV documentaries of nature. And obviously Jacques Cousteau, this is in the late 70s and 80s. Jacques Cousteau was one of those major characters, and one of his series involved Cousteau in the Amazon, and in fact, after he was done filming in the Amazon, he passed through Bogota, through what was going to be my future college when I went to university in 1981, and he visited there for a while. So that's the picture we see here on the bottom left. But obviously that left a pretty major imprint. And when I joined the program, that was in big part because of all that influence about that profession. So I was completely obsessed with this, but at the same time, not knowing how the world works or how to start a career in that, that was the challenge. Where do I go? Who do I talk to? All these things. What do I study? So pretty much in the first or second year of my undergraduate, I heard mentioned that Cousteau had passed through the university, that after he came through, he told several of the students who were there that there were dolphins in the Amazon, in the Colombian Amazon, and that nobody was studying them, and that people should start some research, some studies there. So one of the teachers mentioned that following that advice or prompt from Cousteau, had one of the students had started up a project in the Amazon just a year or two before, and that teacher encouraged me to get in touch with that student, and that student is Fernando Trujillo, who is now world-renowned river dolphin expert. And he was maybe 2 or 3 years ahead of me in the studies in the program. And he was doing his senior thesis. He was going to the Amazon to basically do some prospecting about where to study the dolphins, and well, even to find them, because nobody really knew much about this. And so I was able to eventually get in touch with him, get introduced to him, get in touch with him, and basically I convinced him that he should take me as his research assistant. So I did that for a few years in the breaks between semesters. I went down with him and several other people. This is part of our team, and we would go there and run these boat surveys to count river dolphins. So yeah, we eventually got to see them. And one interesting aspect is that river dolphins are deeply ingrained into the native traditional histories, traditional knowledge. We were staying at a small town, a village called Puerto Nariño, but there were a lot of surrounding indigenous communities, and we would visit them a lot of the times to gather some of that information. And this is a typical wood carving that they make in the area. They have all kinds of legends about the dolphins and their interactions with them. They rely quite a bit on the river, on the aquatic ecosystem for fish, for subsistence, for their livelihood. It's a very sort of aquatic life for them. They have canoes that they are constantly using to go everywhere. So that was an interesting aspect of the work there, was working with the indigenous communities as well. So I did that for a few years, and going back and forth between Bogota and the Amazon. We had to take a flight that was about 2.5 or 3 hour flight to get to the area where we were doing the field work. So meanwhile, I was starting to think about my own project for my thesis and all of that. And right at about the same time, I met Jorge Reynolds, who was involved with the university, even though he wasn't. It was an indirect relationship, but he would often be invited to give presentations at the university, and that's how I got to meet him. But he is an electronics engineer, and he had an interest in developing instruments for a variety of things. So one of them was developing electrocardiogram. Pacemakers for regulating heartbeat and all of that. So he was involved with the medical industry as well. But he was very interested in research with animals. I'm taking the electrocardiograms of animals of all kinds. And so eventually he was getting into measuring the electrocardiograms of more and more exotic animals. And that took him to think that he could take the electrocardiograms of dolphins and whales. And here in the bottom left, we actually went to the Miami's Aquarium to take the electrocardiogram of Lolita. That's Lolita, the killer whale, that it's captive in the Miami's Aquarium. This is probably around 1990 or so. But his grandiose idea was to create a tag for whales that would measure their electrocardiogram and send it via satellite. And this is, again, back in 1990. Bruce Mate was actually just getting started with his studies of just tracking whales. So imagine the idea of not just tracking a whale, but measuring its electrocardiogram and sending the signal over satellites. So his office was a gathering spot for quite a variety of people that were looking to get into studying marine mammals, and he was basically one of the few persons. Fernando Trujillo, the person that I mentioned earlier from the Amazon, he's right here. So he was also someone who would go to Jorge Reynolds' office to be involved in this really exciting idea that he had about building a tag. And so he had a team of people coming up with designs, and we even went to the field to try the tag. We actually never really deployed the tag, but it was an exciting time in terms of possibilities and opportunities. And that actually sent me on a path to look for opportunities to study whales in the Pacific Ocean, in part because Jorge Reynolds knew Roger Payne, and they were going to be doing a project in the Galapagos Islands. And I asked him if he would introduce me to that team that was going to be doing the work there. So he was kind enough to do that, and I ended up joining this expedition on a research vessel called the Odyssey, which was quite famous within the marine mammal research community because it's been in a number of places, including doing a circumglobal navigation back in 2005 or so. But that whole year, which was 1993, 1994, we were working here in the Galapagos and doing some research with sperm whales and doing monthly surveys around the waters of the archipelago. Obviously, over the course of a year, we collected tons of information and data. I'm not going to go into much detail about that, but that eventually became the topic of my PhD dissertation. But that was not to come until later. What I was doing during that year, 1993, 94, was basically assisting with the data collection. And these images that I have here actually are a little bit of again, a sidebar. On one occasion where we very early in the morning spotted killer whales, which you see here from the sailing vessel. Probably it was 6 in the morning or something. And as we approached them, we encountered this freshly dead whale. A Cuvier's beaked whale is a rare species, a deep diving beaked whale. And it was freshly dead, and initially we were not quite sure what was happening. It wasn't until later that we realized that basically the killer whales had killed this animal as its prey, as their prey, because it was a group of killer whales. But not really knowing what was going on, we basically tied up the animal, which you see here on the bottom left, to the side of the sailboat and did a full autopsy or necropsy of the animal and collected a ton of samples from that animal that even today people are using in terms of genetics and stomach contents and the bones and all kinds of things. And then we were basically tossing aside over the side a lot of the remains because it was quite a large animal. And some of these shots are basically blue sharks that came to feed on the carcass that we disposed of after that event. Anyway, as you can imagine, over the course of a year, there were all kinds of really amazing events like this. This was just to give you one example. Some photos of some of the animals, the marine mammals that we were studying there. At the same time, I got to meet Bruce Mate, who came to the Odyssey. This is on board the Odyssey to do some of the first tagging of sperm whales. So that's how I got to meet him and interact with him over that year. And he was there only for a few weeks. But as I told him that I was interested in pursuing a graduate degree, he told me, well, if you ever want to come to Oregon State University, let me know. So a couple of years later, after we wrapped up things in the Galapagos, I wrote to him and I said, you know, I really want to do a PhD, and would you be able to accept me as your student? And also, would you have funding? Because I don't have any of my own funding. And he found ways to fund me and bring me on board. But so, yeah, I ended up doing my PhD in oceanography here at Oregon State University using all the data that we collected over the course of a year. So it was quite a wonderful opportunity to do that work. So if I were to summarize, what some of the things we learned from the research that we were doing in the Galapagos that year, I would say the archipelago has some pretty amazing marine productivity. The waters are very rich because they lie in the path of an ocean current. And what you see here is a satellite image of false color, really, of phytoplankton concentrations, indicating the enrichment that you see in the otherwise fairly low in nutrient and low in biological productivity waters, except around the islands, where that productivity essentially fuels a very rich food chain that attracts a lot of marine mammals. And this is the last slide I'm going to show on that topic. But if I were to summarize again, what the results of that PhD research that I did was to essentially identify the most common species. There were about 9 or 10 species that were very common, but they all had very distinct distribution patterns. And through the analysis that I did during my PhD, the idea was to unravel their habitat preferences for each of those students. So each of these curves that we see here corresponds to one particular species. And I'm probably not going to go into much more detail than that unless someone is interested. But the idea is that the abundance of those species along some environmental gradient, so this axis 1 is an environmental gradient. It can be marine productivity, or it could be temperature, some kind of gradient. The abundance is sorted out such that some species peak at one end of the curve or the axis, some species peak somewhere in the middle, and so forth. So that was the idea, was to unravel or figure out the structure of that community. There's actually one more slide before I pause. And that slide is to say that after I wrapped up my PhD and so forth, I really didn't do much more work in the Galapagos, except that very recently, this is as of last week, a new series of important marine mammal areas in the Pacific Ocean were announced, and among them was the Galapagos archipelago, and that was the outcome of essentially being able to synthesize a lot of the information that I collected and analyzed during my PhD. It was used to inform the delineation, identification of this important marine mammal area, which is now part of the global Marine Mammal Protected Areas Task Force. And this can be found online it's an atlas that it can be found online. So maybe I should stop here and see if there's any questions, because I haven't been really keeping track of that.
I have a ton of questions.
Oh my goodness. Okay, let me stop the sharing for a moment and I'll let you help me with the questions then.
Excellent, excellent. Well, we're going to give everybody a little nudge to post their own questions, but I have questions. So first, I didn't realize, and obviously if Bruce went there specifically for that, just didn't realize the Galapagos had such an abundance of sperm whales there. I didn't realize that was kind of a hot spot. Is there a seasonality for that, or are they kind of year round?
So that was actually part of the question that we wanted to, well, that Bruce wanted to do, because figuring out that seasonality can be hard. But with satellite tagging, you could see animals, for instance, moving away and going toward the poles, because the Galapagos are right on the equator. So their presence is generally year-round, but really there hasn't been, other than that year that we were there, there hasn't been a lot of work to figure out if they are there year-round or not. We did find them year-round, but it was clear that there was groups that were moving in and out. So they were constantly fluxing and coming in and out. So the Galapagos were actually one of the original whaling grounds during the 19th century whaling. And basically they started in the 1830s, and by 1950 they had pretty much decimated the population there. So it's been on this slow recover, but the part of the thing is with the sperm whales, because they have such complex social organization and family units and clans and all these things, it's taking them a long time to recover. And some of the modern work that Hal Whitehead has done has all been based on studying whales around the sperm whales around the Galapagos, and it's quite fascinating, that whole story about how the population has been recovering, and understanding the social structure of those groups has all been through Hal Whitehead's work around the Galapagos as well.
Wow. I have one more question about the Galapagos, since Bruce was there tagging, and it looked like quite a young Daniel in that picture. So how long, not to get into all of the details of all the tagging, I think a lot of people are kind of familiar, most of the people on tonight are a little bit familiar with the enormous amount of data that can be collected from tags today, but how long would those GPS tags track back then?
Yeah, so turns out that none of the tags worked. So this was in 1993. Bruce had just started deploying tags on whales in the late 80s and getting them to work. They were quite different from what they are now. They were larger, and quite in a number of ways they work differently. But turns out that because sperm whales dive deeply, and he knew that, and he designed the tags in a way that they would be resistant to the pressure during the deep dives. But apparently the tags had some crystals inside of them that as part of the electronics, and sperm whales make some really loud vocalizations that apparently were quite in the frequency of the crystals, so it resonated them and basically disabled those tags. So even though we had a successful tagging operation while he was down there, unfortunately, it was a tough lesson to learn that none of those tags work. So he kept working and developing the tags, and eventually he went to the Gulf of Mexico and successfully tagged quite a few sperm whales, and we have lots of really wonderful tagging data for sperm whales in the Gulf of Mexico, but we have not been back to the Galapagos. So the question still remains about where are these animals that go to the Galapagos? Do they come from? Do they come from the northern hemisphere? Do they come from the southern hemisphere? We're still not quite sure.
Anybody out there that's looking for a PhD project might talk to Daniel about that.
Exactly.
I have a couple questions of your earlier years, but Tamara is asking about what the 9 species were, and I think she's curious about your graph. I don't know if you want to turn that back on or not, but
yeah. Let's see if I can do that.
I turned that back on. Share the slide again.
Trying to follow my steps here. So I need to share first the PowerPoint.
And it looks like, I don't, is Raphael a family member?
That's
He's got
my
his,
dad and my mom.
oh my gosh.
Tuned in. Yeah. So let's see. Here in the maps, there's 3 maps. There's 2 species in the first map: spotted pantropical spotted dolphins and spinner dolphins. And the black points show where we had locations where we saw them during the surveys. The black points in the middle map show the short-beaked common dolphin, and you can see that it has a very different distribution pattern compared to the other 2 dolphin species. And then here, you see the distribution of sperm whales, which is somewhat similar to that one of the common dolphin. Let's see, so there's 4 species there. There was rhesus dolphins, short-fin pilot whales, bottlenose dolphins, of course, they're quite common very near shore. And Brutus whales. Brutus whales are a tropical baleen whale that is actually quite common around the Galapagos. So those were the primary species that we encountered. We saw a number of other species, but we didn't have enough data to do any analysis of them.
Excellent, thank you. So Raphael has his hand up. We could allow to talk. It's your relative. I'm going to let you decide. Or let's see. Oh, he just took his hand down. I don't know if he wanted to say something, ask a question.
Let me see.
Okay. If he has a question, there's certain guests we could allow in. I'm going to have Nikki post your question in the Q&A, please. It looks like there's something in the chat. And while we're waiting on that, there's some other information. But I had a question about, you traveled a lot, you made a lot of great connections. It looks like, you had to be quite ambitious, because I think if everybody thinks back to, I'm not going to, I don't know your age, but you could tell from the pictures, it was a little bit ago, it was a minute ago or so. There wasn't internet and easy email, I think.
No, exactly.
And,
so
and
that
you
was
were
back
in a
the
remote
time.
area, kind of, right? In
Yeah,
Colombia?
exactly. Yeah. So, we had to rely on the regular mail and writing letters the old-fashioned way, and waiting months to hear a response. Email came on board, at least for me, in the mid-90s. That's when I started using email. But yeah, back then you had to rely on the regular mail, and especially for some of these remote areas, it would take months to get a response or anything. So yeah, in terms of trying to contact people, especially overseas, that was quite the challenge. And sometimes you'd send a lot of letters and, not hear from several people and hear from some. So yeah, it was different times. Yeah, so I'm 52, about to turn 53. So
I'm familiar with that area, that time frame. And I mean, yeah, you were doing some pretty remote work. And so when you boarded the Odyssey, I read a story, so I'm going to give you the little leading question. Did you just run down to the dock and meet them, or had they been traveling, or did you write a letter to them and hope they threw a message in a bottle back, or how did that work?
So going back to your point about being ambitious, I'm not sure if ambitious is what I would call it, but I would call it passionate and full of dreams and persistent. So I mentioned this fellow Jorge Reynolds that I met in Colombia. He was quite influential and quite well connected and well known, and I think many people benefited from his contacts, his resources, all of those things. So I mentioned he was friends of Roger Payne, and through him we learned that the Odyssey was sailing from Key West in Florida through the Panama Canal on its way to do this year-long expedition in the Galapagos. And it was while the ship was in Panama, which is right next door to Colombia, that Jorge said, "Oh, I'm going to meet with Roger." And I hope that most people here will know who Roger Payne is, but he was a very famous marine biologist in the 1970s and 80s who discovered and described the song of the humpback whale. So yeah, he was very well known globally, and Roger came to Colombia as well. So through some of those contacts, basically I told the person that I work with, Jorge Reynolds, I said, please, please, please take me with you to Panama. I know that you're going to meet with Roger there in Panama. Please take me there and let me meet them. I really want to get on that ship and go to the Galapagos. And he initially was no, 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 no. And finally, I insisted so much. And I have relatives. My aunt lives in Panama. And I said, listen, I can stay with her. I'm not going to, bother you in any way. Just introduce me. And he finally, I probably just pestered him so much that he finally agreed. And he introduced me. He left like a day or two later. He was just there to say hello. It was kind of like a business meeting or something. Hello, goodbye, it's done. So I ended up staying in Panama for about a week or so while the boat was going through some work at the marina. And basically I would show up every day, and the person there who was in charge Ian Care, who's also quite well known, he would say, no, we can't take you with us, but if you want to help us, we have lots of paint chipping and rust removing and all kinds of work that needs to be done on this sailboat, so you're welcome to help us. So I showed up every day during that week or week and a half, something like that, and work with them. And during that time was able to sort of basically build a rapport and get friendly with them. And then the last day, as they were basically leaving and from the dock saying goodbye, he said, okay, get on board, you can come with us. But the moment we get to the Galapagos, which is it's going to be like about 7 or 10 days sail, you have to get off, and don't ask me for money or anything to get back home. You have to figure out how to get back home, but under those conditions, you can come with us because we have a complement of scientists, very famous scientists that are coming from Europe to do some work on the sperm whales, and there's no room for you, so you have to get off. And I said, yes, I'll go for that, sure. As it turns out, those scientists never showed up because of some issue, and that's how I got to stay there for the whole time. So basically, after we arrived, he said, well, maybe you can stay a week or two, and then maybe you can stay a month or two. And then I ended up staying for a whole year.
Wow, that's fantastic. You were valuable. Yeah, that's great. And that's where you met Roger.
Exactly.
Excellent. So Nikki has asked a really good question, but I'm going to opt to hold off because it's a really great question that I think I want to end with. But I do want to get in Tamara's other question. Can you share what the river dolphin was in the Amazon that you were studying?
So there's 2 species of river dolphins in the Amazon. One of them is the boto, or Inia. Is the pink river dolphin. It's a prehistoric, very large pink river dolphin. The other one is called the tucuchi, or gray dolphins, they also call it. It's the genus is Sotalia, and it's actually a marine dolphin that evolved to migrate upriver. So unlike Inia, which is a dolphin that originated and evolved in freshwater systems, Sotalia is a very recent in evolutionary time in terms of colonizing. They were originally from the coastal marine environment, but they colonized the Amazon, and now you can find them. It's actually it's been separated species taxonomically. It's not the same. There is a coastal marine species that's now known as the Guiana dolphin, and then the river form is known as the tucuchi, and they're entirely separate species, even though morphologically they're almost identical.
And I'll say, Tamara, we'll take a look at those during class, or you should look them up. They're very interesting. River dolphins look a little bit unique compared to regular dolphins, or oceanic dolphins, I should say. So yeah. Let's get you back to it. Let's hear some more.
Good.
All right.
Let's make some more progress here. So after I was done with my PhD here at Oregon State University, I went to work for NOAA for 11 years in Monterey. So this is actually Pacific Grove.
Share your screen, please.
I'm sorry.
Sorry. Thanks.
That would help, right?
Maybe you could be descriptive with your language, but if you have pictures.
A lot of imagination there.
Swap it again.
Swap them again, right?
Yep. There we go. Yeah.
Great. So yeah, but this was back in 2002, 2003, after I graduated from my PhD.
It swapped back. Can you swap again? Sorry, swap display.
No worries.
It didn't. No, interesting. It's still showing presenter.
Let me do something else here.
Oh, both times.
Let me share first and then do the presentation mode. I think that helps. What do you see now?
There we go. Perfect.
Oh, working. Great.
Yep. Thanks.
So some of you will be familiar with this building, but it used to be a Cold War era bunker by the Navy, and then in the 1970s it was taken over by, or 1980s maybe, by NOAA, Southwest Fisheries Science Center. Pretty small facility, but basically I went there to do, and right next to a golf course, the Pacific Grove public course. And that particular center specialized on studying the oceanography and ocean climate. And I was interested in going deeper into my studies of oceanography. So I went there to do a postdoc for 1 year, but 1 year ended up becoming 11 years. So I was based out of there for all that time, and basically until 2013, when I moved back to Oregon State University to take my current position as a professor. But this was a really wonderful time in Monterey, and I wasn't necessarily working with marine mammals as much as I had been in the past, especially not doing field work. But this center was involved, which, by the way, it was called the Pacific Fisheries Environmental Lab. It was part of a project called the Tagging of Pacific Pelagics, which I think now has been renamed as the Tagging of Pacific Predators or something like that. But the idea was to put all these electronic tags on quite a lot of species, including sharks and turtles and seabirds and elephant seals and so forth, and track them through their movements in the Pacific Ocean. And it was quite, again, the opportunity to be part of that. And that's how I started really working on tagging data and doing analysis, integrating the tagging data with the environmental data, and coming up with how we understand the distribution of movements of these species. And that way help with better protection as we better understand their spatial patterns of distribution. So I did that for I mentioned 11 years. And then I came to Oregon State. And that was to work with Bruce Mate. And as I mentioned, he was the pioneer and developer of these long duration satellite tags that allow us to track whales for periods of up to a year, sometimes more, sometimes less. The specific duration varies quite a bit, but it uses a system. It's not GPS, it's a different satellite system called Argos. It doesn't have the same accuracy as GPS, but it's pretty good. And in this photo here, we see a fin whale. These tags are basically completely implantable because some of the early lessons that Bruce learned in the process is that tags that were mounted on the surface of the whale would not stay on for very long, and they caused all kinds of problems. And they need to be anchored and attached, and so they need to be implanted. So they're invasive, but even 20, 30 years later is the only way that we have to tag whales for track whales for long distances. There's other attachment approaches and other types of tags that give us some excellent information, and we use them as well for short-term tracking, but for long duration, beyond more than, say, a few weeks or months or a year, this is the only method that has worked. and it's being used quite widely, as we will see later. So I'm actually going to show you here tag deployment operation. This is a few years ago in the Strait of Juan de Fuca in Washington, and there's 2 humpback whales that we will see up here to the right. There's one animal here, and we'll see another animal that's coming up next. What you see here at the tip, this is the tag that's implanted on the whale. The long red part is part of the launcher. It's an air-powered. You see a scuba tank here at the bottom. So it's powered through air, compressed air, and that is what launches the tag into the whale. So we're going to watch that operation. We have we have this bow pulpit. We call it the bow pulpit, which allows us to be in a vantage point so that we can find. Putting a tag on a whale is quite a precise operation. We want to put the tag in just the right spot so that we try to mitigate harm to the animal as much as we can. But as I mentioned, these tags are invasive, but also so that the tag is exposed to the surface, because the tags are only able to transmit when the animal is at the surface. They do not work underwater. So let's watch that. We select the biopsy sample at the same time. More about it in a moment, but you see the 2 events happening at the same time. So that's it. That happens over just a few seconds if everything goes well. And the tag has a carrier. Once the tag is implanted on the whale, the carrier falls off. The biopsy dart that you saw, the arrow just went flying. So at that point, we pull back and go back and pick up the carrier as well as the biopsy arrow. And with that piece of skin and blubber that we take from that biopsy, we can learn about the sex of the animal. We learn about the genetics, the haplotypes, the genotype of the whale. We work with Scott Baker here, who does all the genetics work for us. We are doing studies of stable isotopes to learn a bit more about the feeding ecology through the analysis of, as I mentioned, stable isotopes in the skin sample. We can do hormone analysis, fatty acid analysis, so quite a lot of analysis that we can do that. And I have students that are working on using those samples to learn more about the animals that we have to. So it's a nice way of combining a variety of techniques. It's not just the tagging through the biopsy work. We get to learn quite a bit. And as you see here, we have Craig Hazlett here. Some of you may know him. He's our photographer. So his job is just to take photos of the whale. And then we, in the case of humpback whales, we put them on Happy Whale, and we pretty much immediately can know which animal, because most of the humpback whales in the North Pacific are now known and cataloged, and they're on Happy Whale. So we know which animal we tagged. So again, quite a lot of synergy in terms of data uses. Oops. All right. There. So stepping back a little bit, Bruce started tagging humpback whales in Hawaii in 1995, and these are some of the early tracks that he collected. And are you able to see my screen still correctly?
Yep, all good.
Excellent. So this is from a study that Bruce published in the 2000s, but this is data from 1995 to 2000 of the tagging work that he did in Hawaii. So this was the first time that we were actually seeing whale migrations, where they in every detail about where they went. And great discovery, even though this is just a few animals, but you can see that we knew that this is back in the mid 1990s and early 2000s. We knew that most whales in Hawaii came from Southeast Alaska here, but through this tagging work, we found that some animals went to the Eastern Aleutian Islands. This is Unimak Pass, a very famous region in the Eastern Aleutian Islands. And in the case of one whale, it went to the same area, but that it continued on westward all the way through the Aleutian chain. This is Kamchatka Peninsula in Russia. And basically that's where the tag stopped. So that was quite new information. And that's how we got to learn how they move between these areas and between feeding and breeding areas. So that was tagging done in Hawaii. Similarly, in 1998, there was some tagging work done at Socorro Island in Mexico. So this is a different breeding area. Hawaii is one of the primary breeding areas for about half of the humpback whale population in the North Pacific, which is about 20,000 animals. And 10,000 of them roughly go to Hawaii, and the others go to several other areas, but one of them is the offshore islands of Revillagigedo, of which Socorro is the main one. So he tagged some animals there, and these are the routes they took. So again, kind of like for the first time, describing those patterns of movements, including within Mexico, which is still the topic, quite a lot of work being done to try to understand, because there's a lot of different populations that winter in Mexico, and we're trying to understand how to separate them, because they tend to overlap quite a bit, even though we know that they are distinct and they actually have different levels of exposure to human risk and conservation. So it's still quite the topic of active research. So that was the work through the late 2000s. And then more recently, with additional work, what I'm going to do here is play you an animation. It's probably about a minute and a half, something like that, where we will see now the movements of all the whales that we have tagged and tracked in the North Pacific. So let's watch that. Again, tagging Hawaii first, and this is done in February. You can see the month of the year at the top here, and also the year day. So still in February, animals starting their migration toward the feeding areas, as well as the other breeding area that I mentioned in Socorro, in the Revillagigedos, and migrating to the feeding areas, kind of like fireworks. So they arrive to the feeding areas. You can see some of them cross paths and do all kinds of really interesting uses of the environment. In this one case, one whale went into the Bering Sea here. And now we're in July, which is the feeding season, and we've done some tagging in California and Washington and Oregon and Southeast Alaska, and here in Unimak Pass as well. So now you start seeing the tracks of the animals that we tagged during the feeding season in those areas, and how they're moving both within the feeding season, the feeding areas, and in a moment we'll start to see their migration toward the breeding areas. So in this case, we see an animal or two or three moving toward Mexico, animals moving from Southeast Alaska back to Hawaii. Some animals from Washington also starting to move to Hawaii and Mexico. So quite complex, quite a complex picture there. This is about the tracks of about 140 animals here. More recently, beyond doing the work that we do as a lab, as a group, there's been a lot more collaboration between different researchers that are using the same methodology and the same technology to try to put a more global picture together. And earlier this year, we published a report called Protecting Blue Corridors, Challenges and Solutions for Migratory Whales Navigating International and National Seas. This was led by Chris Johnson at WWF and a variety of people, including University of California, Santa Cruz, and many other contributors. But the idea was to bring together the scientific community tagging whales around the world and tried to put all those tracks together. So over 50 research groups contributed their satellite tracking data. We put together over 900 tracks over a period of 30 years. And basically, the key messages of the report is that protecting whales has benefits for nature as well as people. Whales play important ecological roles that we're just starting to understand and really define and make them justice, do them justice. And what we call blue corridors are those areas that essentially you could consider them like highways, migratory highways, where a lot of these whales are circulating through. And maybe from the previous animation that we got to see, you get a sense of some of these areas where a lot of their migratory areas are concentrated. Yeah, whales are an indicator of ocean health. And at the same time, they are facing growing threats. You know, the threat of hunting, fortunately, has stopped, but there's a lot of new threats, especially in the high seas where there's no protection, as opposed to in national waters, the jurisdictional waters of many countries that have implemented laws and policies to protect whales. It only goes to the national waters, but as we saw, these animals are swimming across ocean basins, and a lot of the high seas are unprotected, and yet there's increasing shipping traffic, increasing pollution, increasing ocean noise, increasing fishing pressure. So the ability to precisely map those corridors, those blue corridors, is pretty much the next step of where we need to take things through enhanced cooperation, which is what we were able to do, putting together this. So this is the global map of whale movements and migrations, those blue corridors, through collaboration with a lot of colleagues that contributed their tracking data for quite a few species. We're looking at fin whales, bowhead whales, gray whales, North Atlantic right whales, South Atlantic right whales, humpback whales all around the world, sperm whales, blue whales. So it's quite exciting and quite impressive to, for the first time, have this kind of global view of whale migration. And now we continue this work by focusing by region, because we understand that it's really exciting and really cool to see this and to be able to map the various layers of threats, including climate change. I mentioned fisheries bycatch and overfishing of the food resource, and ghost nets and shipping, and all those layers of various human activities that threaten these animals today. But then a lot of these things need more concerted regional solutions. So the next step with that, the work that we're doing with WWF, is to go ocean basin by ocean basin and try to identify the problems a little more closely. And that's all through the combination of the satellite data that we have been collecting over the last 30 years with these global layers of uses of humans of the ocean. So, for example, here in the Eastern Pacific, which is what we're working right now, some of you may have heard of the Great Pacific Garbage Patch. So we are able to match where to map where that patch is in relation to the migratory areas, the migratory corridors. There's areas of exploitation of minerals and all that. So that's what we're working on at the moment. This is the latest initiative that we are undertaking today. Maybe see, yeah, I have just this video that maybe will be kind of like the last slide that I have. I may have one or two more, but let's just watch this because it's kind of like wrapping up a little bit of that Blue Corridors effort. It's quite exciting. So let's just watch this.
Every year, whales make some of the longest migrations on Earth, swimming across oceans to feed, mate, and give birth. These blue corridors are migration superhighways, critical for their survival. However, these journeys are becoming more dangerous to navigate, with growing threats from industrial fishing and ghost nets, ship strikes, underwater noise, plastic pollution, and climate change. Because of these dangers, some of our great whales, which have survived for millions of years, could go extinct within our lifetimes. Science shows whales help fertilize our ocean and fight climate change. Throughout its life, one whale captures the same amount of carbon as 1,000 trees. We are calling for a new conservation approach to protect whales in their critical habitats so they can thrive for years to come. Together, better managing and protecting blue corridors can help safeguard whales, our ocean, and ourselves.
Yeah, so that was the video that was put together by WWF for the launch of the report. I'm going to stop sharing my screen, but I will say that the idea with that report was actually, and we intentionally did this, we didn't produce that necessarily as a scientific publication like most of the work we do. It was put together as a report with a series of sections and chapters that were tailored to policymakers. So in the last year or two, there has been a lot of work at the United Nations with what's known as an initiative known as Areas Beyond National Jurisdiction. So these are the high seas where currently there is no policy or management, and it requires quite a bit of human cooperation within the United Nations, essentially, to be able to produce a globally consistent policy that all nations can get behind to be able to implement protective measures that all nations can agree to follow in their activities that happen in the high seas. So that was the idea beyond that report, that project, was to directly reach the policymakers. So we actually worked with WWF policy people who are quite involved in that to help tailor the message and to produce information that was going to be directly useful. The United Nations, the various units within the United Nations meet annually during those COP meetings, the Conference of the Parties. And last year and again in the next year, we continue that effort in terms of delivering that message directly to them. So that's, for the most part, the material that I prepared for you today. But yeah, maybe we can continue some questions, and maybe I'll come up with more, some slides or more material if necessary.
Absolutely. We have a couple of questions, but so I posted a link to the Protecting Blue Corridors report. That was fascinating. Thank you for sharing that. And the video was really great and strong one. I think hopefully it gets to the right people. We've more and more research is kind of pulling together this holistic look at animals, and it's really great to put it all together, right? This work and the tagging data that you've been working on for decades, but also with things like Ari Friedlander's work, looking at their feeding capabilities and the density of the prey. A wide variety of people. You guys obviously collaborated with a number of people here, but it's really interesting to see what the world's marine mammal community is doing, other communities as well, but to show the value and the importance of whales in our ecosystem and environment. And I think there was a publication recently about actually putting the dollar value on a whale and how it relates not just to our environment, but to our economy as well, right?
Yeah, that is a really interesting exercise that was done in the context of the, I believe, International Monetary Fund. So an economist did a whole study to be able to come up with what is the cost, how much does a whale cost? And they came up with this number of about $2 million. It necessarily doesn't have to be too accurate to just tell you that these animals are more worth alive than death, because it's only through their activities while they are alive that they're able to accrue that value, in the sense that they're sinks for carbon, for example, and they're fertilizing the ocean and moving matter and so forth. Not to mention the incredible explosion of whale watching activities, and how humanity really is becoming more connected with nature by taking one whale watch trip at a time, and I know that you guys down in Southern California are quite involved in all kinds of fantastic whale watching activities, and it's quite vibrant, but it's so rewarding to see so much more of the general public become illustrated and informed on whales, and wanting to share that. They're such magnificent, incredible animals, that to be able to go see them in the wild is just unparalleled experience, and it's so exciting. I've been working with whales for decades, and every time I see one, I get so excited. You just can't contain the excitement, because they're just so magnificent, really. So
it's true. I know there's a lot of controversy around some unethical whale watching practices and things, close approach and encroaching, and injuries and stress, but I think it's hard to balance, or it's important to balance that, because it's also very valuable, and it's we're getting people out in the wild and seeing these animals, and I think it's that connection that's going to help them take the next step to making a difference, versus reading about it or seeing it on TV. But to when they feel that connection to the animals and then to the ocean, I think it connects them then to nature and protecting our environment, really.
yeah, it certainly has that effect, that sort of multiplier effect, in terms of reaching people that normally would not have the opportunity to do this. And yeah, we just don't really know how far-reaching these impacts can go within society. So it's exciting times, and at the same time, troubling times, because of all the increasing pressures and threats. So yeah, there's always this delicate dance and balance.
We have a couple of questions here that I don't want to overlook. Tamara had a couple more. She's got some great questions tonight, but I'm going to lump a couple of these together. She asked about the tagging, and does it hurt when you tag a whale? And in regards to the humpback whales, do they choose, like, how, what's the, she says, how do they choose where they go? Hawaii, Mexico? Is it based on their past? Are they returning to a natal ground? That kind of thing.
Sure. So let's tackle the last one first, regarding choosing between Hawaii and Mexico. So there's this idea that because the mothers tend to always go to the same feeding area year after year, and then to the same breeding area, when they have a new calf, a new baby, the calf will learn the same route and generally follow that. So that's called female-driven philopatry. And for the most part, animals tend to follow the same route. But what we're seeing, in part through this work, tagging, but also through photo identification, is that more and more animals are actually switching. So we have several cases of whales that we have photo identified in Hawaii, and then they have been seen in Mexico, for example. So, and there's other colleagues that have found the same thing. So there's this idea that perhaps as the populations continue to grow, there's a little bit of more exploration, and animals undertaking longer migrations or going different places, especially younger males probably are the ones that are doing that. So definitely there is some degree of exchange, and that's what keeps these populations to not be completely isolated, because that just means that there's some genetic change going on. So even though they have very distinct genetic makeup, all these different populations, at the same time they do share some of the same genes, some of the same haplotypes. So yeah, there is a little bit of exchange going on between breeding grounds. So that's kind of an exciting, interesting result that we're seeing.
Yeah.
So to get to the first question about does it hurt them, I would say, what do you think? And the reason why I say that is not to be facetious, but it's because actually we don't really know how whales experience pain. So I don't think it takes any genius to know that animals experience pain just like we do, but exactly how they experience it, especially whales, which are highly derived organisms compared to, say, terrestrial mammals. So a lot of terrestrial mammals have relatively thinner skins and very, very different skins or hides or whatever, basically no blubber to speak of, and nervous systems that are very different. So whether they experience pain in the way that us humans think of it or not, that's harder to tell. It's one of those really hard things to really be able to measure scientifically, and that's where, you know, that's where we come from as scientists, is trying to weigh the benefits and the drawbacks of those various methodologies. So undoubtedly, and from watching and being involved in these operations, I can tell that my sense is that they do experience some pain. But what we see, for example, when we collect a biopsy, just a biopsy sample, which is a tiny little piece of skin, their level of reaction is quite the same as when we tag them. So with, you know, those limited ways that we have of assessing whether an animal may be in distress or not, what we see is that they return to the regular activity. Now, that doesn't mean that they're not experiencing pain anymore. It's just that we don't really know. So what I have to say about that is that we come at these approaches over decades of development of techniques to tag and track animals that is the realm of wildlife biology. So tagging animals is not something that we just do with whales. People are doing the same with African animals and birds and terrestrial animals, you know, reindeer or cougars, mountain lions, whatever, all over the world. They're using various approaches to tagging that are adapted to the specific animal. So we're not doing this in isolation. It's kind of like what the wildlife research community is currently accepted technologies and techniques. And so we have to go through a lot of very rigorous approvals and permits, authorizations, and oversight by committees that examine our use of animals. We have to have IACOC, a protocol for doing our work with animals. So these protocols are regularly assessed every year, and they are approved. All that to say that our methods are generally approved by the wider community. We participate in a variety of, in addition to NOAA gives us their own permit, which is quite rigorous to obtain. Then we also obtain our IACOC protocol, but we are also involved in other organizations that likewise assess our methods, and we're constantly reviewing them and constantly improving them to mitigate what I think it's undeniable that these invasive tags have some level of impact on the animals. But what we're seeing is essentially a changeover, a rapid changeover from where we came from, which the driver was, we're interested in learning this information because we want to protect the population. We want to generate information that will help us for the benefit of improving the conservation status of the population. That has been traditionally the approach by wildlife biologists and by management agencies. So if you overimpose on that what's been happening in the last decade or so is with the rise essentially of social media and the general public interest in animals, is this idea of individual animal welfare. So we're actually coming at this from opposite end of the spectrum, where the welfare of individual animals also matters. So as scientists, we also have to be receptive and understand that perspective, even though for us we are very focused on certain aspects that we know how to do well, and we put all our efforts in that, and traditionally have been doing that. But as this new level of interest in the welfare of the animals is coming on board, there's a stronger drive to mitigate and improve and change some of these approaches. So change is definitely in the air in the way we do things, but all that to say that basically we have been working and there's certain paradigm, and that paradigm is changing. But by no means, I would say this is driven in a high degree by the people who care about whales, because they're so charismatic, because so many people get to see them and see all this. But nevertheless, people maybe are less interested in how other wildlife, like let's say raccoons or beavers or whatever your backyard animal is, what approaches are being used. They're maybe less visible. So when it comes to whales, everything's a little bit of a higher stake because people have that sort of attachment to the animals. And we do too, but we come from very different angles. And it's important to understand all these perspectives. And yeah, we're definitely working to improving all of these things.
That was an awesome answer. Thank you very much for taking the time to really spell that out and talk about the different, the development and of course all of the different authorizations, but the awareness and concern that you guys have on the different angles and effects. We try to wrap up by 8:30. But I have one more question that I've been holding. I think Nikki's still here because she just posted another question, but I want to get this one question in. I want you to answer that question. Then, depending on if you're willing, whoever wants to stay, but we have a couple more questions that are coming in, but also we can drop off. But Nikki's early question was, of course, thank you for sharing your fascinating story. She did ask about your most exciting experiences during your early expeditions, but I like this question to end on. What do you think your personal most definitive characteristic that got you where you are today and helped with your professional success?
So thank you, Nikki, for those questions. I know Nikki very well. She was a student here at Oregon State and through Greece. She's from Greece originally, but she's a wonderful friend of mine and very dear to me. And so actually, to some degree, the answers to your questions today, I kind of covered them in that probably one of the most exciting and just kind of outlandish situations was when I mentioned that killer whale kill of Cuvier's beaked whale in the high seas or far away from shore in the Galapagos while we were working there. So it's just kind of like you wake up one morning and this flat calm is very, very calm. And we see these killer whales at a distance. Oh, wow, let's just go and document them. And then, they kind of avoided us and they left, but then we just found this dead animal in there. And it's just like how we proceeded, what do we do here? And, do we just leave this animal floating out there? Or do we we didn't even know what species it was when we found it. It was a juvenile, and only later through the analysis of the bones and various other analysis, we were able to identify the species. But just that whole thing just seems so out of this world to be able to experience that. And, it took us probably the most of 2 days at sea to be able to do that, because we were underway and, in a sailboat doing this work. So it was kind of far out there, really. And as far as the other question about a defining characteristic, I would also say that I kind of touched on that when I said that I would call it ambition, but definitely having these dreams from early on that I was kind of determined to pursue. And even though they couldn't seem more remote, you know, as you imagine someone growing up in a city up in the Andes, far away from everything, and in a big city, you know, Bogota is a city of over 8 million people, and not really having any exposure to the ocean or anything like that. So I would say just kind of like always that passion for animals and wildlife, and then just the persistence, having the persistence and sticking with it through. You have to work really hard. That's probably the persistent aspect of that. So having the passion, the dream, and the persistence to stick with it through high and lows.
Yeah, passion. You can have the passion, but unless you have that drive, that persistence, you almost need them hand in hand, right? Because you can also be persistent, but not necessarily know, you know, what you're after. You're just you know, you just keep going. But tenacious, Kathy, I think, called it too.
That's
but
a great word. I love that. Yeah.
Is the new tenacious D here? No. Thank you. I'll quickly, for those of you that are dropping, we wish you guys all a very happy holidays. We will not have a deeper dive webinar in December, but you can look for information about our deeper dive webinar series or last Tuesday of the month lectures starting again in January. And thank you very much. And I will say, while everybody's here, thank you so much, Daniel. This was a really, really fun evening to hear about your story, your history, and how you got where you are. I think it was really inspirational for a lot of people still pursuing their future and their interests, young or old. They might still be pursuing a lot of passions. And I think that this was really great. I hope you'll take a second to read some of the chat accolades that you're getting and are well deserved.
Thanks, everyone.
And if you have a moment, we can have a couple other questions, but I'll leave that up to you because it is getting late.
Yes.
So Carlia went back to the humpback whales, and you talked about them switching Hawaii to Mexico.
Yeah.
You picked that up, and she just wondered if there was a reason that you're seeing mating, food, she asked, but.
Right. Well, there's a little bit of that notion that with recent climate changes in the North Pacific, and we had the marine heat wave in the mid-2000s, 2010s, that really disturbed or disrupted the marine ecosystem. It is generally understood that during those years, whales had to shift their migratory patterns. You know, they were not going to Hawaii in the same numbers. They were not being seen in Mexico and other parts of Central America in the same numbers. So where did they go and what? So there may be some aspects related to finding food in other places that may be driving that. But my own guess is that some of these animals that are switching back and forth are more related to, this is common behavior, I would say, in many mammal populations, where the younger males of expanding population, because the whale populations are growing for the most part, not all of them, but certainly the North Pacific humpback whales are a success story for the most part, is that as the population grows, there's more density. So there's this process of density dependence, where the more animals are in a certain area and they're occupying sort of the prime habitat, then the new animals have to kind of like find this marginal areas around the periphery where it's not so good, so they might as well, I'm just going to go over here and see what I can find, you know. So maybe there's some aspect of that sort of density dependence, roaming around. Males, of course, may have an interest in exploring other areas and other mating opportunities, so maybe there's some of that going on. But it's something to definitely needs more research, more. It's a really cool question.
Yeah, that's interesting. I didn't realize we had so many sightings of shared, breeding grounds. I really thought most of those Hawaiian humpbacks were still going up to Alaska and not really and returning there. So that was interesting, and that's what all that tagging data brings us. Really, is that really fine scale information. Nikki did have another question. She said you've incorporated more sensors into the satellite tags, like accelerometers, which is great, really pulling in a lot of other additional information.
Right.
She asked if there's any plans to incorporate any acoustic sensors in the future. She recognizes that acoustic data are heavier to transmit remotely.
Yeah. Thanks for that question, Nikki. So for the longest time, these long duration implant tags that we have been using, they only have had one functionality, which is to relay the location of the animal. Part of that is because in order for them to have long duration, they're not archival where you recover the tag and then you download your data. It's all transmitted via the Argos satellite system, and that system has some severe bandwidth limitations. So for the longest time, we've only been using these tags for tracking the movements. But there's been several developments, definitely the fact that through cell phones and all of that, a lot of the components that are in cell phones have become cheaper and smaller. So in the last few years, we have been incorporating some of those sensors into our tags. So our current tag, actually, in addition to being able to relay the location of the whale, we are able to measure the depth, the maximum depth of each dive. And also through the accelerometers, we are able to identify feeding events of certain species, not all species of whales, but at least the rorqual whales, like blue whales, fin whales, humpback whales, that use the lunge feeding method to feed. They leave a very particular stereotypical signature in the accelerometer data that then we can use to, we have an algorithm as part of the software in the tag that just is constantly monitoring the accelerometer signal. And if it sees the stereotypical feeding, lunge feeding, then it counts that as one lunge feeding. So then what we get is the number of lunge feeding events that the tag measured. So that information, the duration of the dive, the depth of the dive, and the number of feeding events per dive they get packaged. And then the second development is that there's been some advances in data packaging through some creative algorithms and modern computing that essentially allow us to package that information into bite-sized messages, literally, that get transmitted through the Argos. You know, we're basically looking at this tag that is diameter is only about 2 centimeters in diameter. It's sending a signal through an antenna, through the atmosphere, through a satellite that are about 1,000 kilometers overhead, and picking up this. So it's quite limited into what you can do transmitting data, as opposed to the more fully featured, very sophisticated, fancy archival tags like D tags or CATs or suction cup tags that people recover, and essentially they're a hard drive that store all the information. In this case, we're just picking up the most important events and packaging them for transmission. So that is the latest, and we're quite excited with that new tag. It's giving us some fantastic results about information that we didn't have before, and we're able to gather that information months on end. So for the first time, we are extending what we are able to learn about how much a whale is feeding, not just over the course of 6 hours or so, which is the typical deployment of a suction cup tag, but over the course of a week, over the course of a month, over the course of many months. So how are they changing their feeding patterns and feeding behaviors over time as they migrate, as they dive through different parts of the ocean? So it's super, super exciting. As far as acoustic sensors, that is the next frontier. Definitely is on our wish list to be able to also include some kind of acoustic sensor. It probably will require some packaging, similar packaging. Acoustic data is extremely dense, as Nikki mentioned, heavy, because it's all these frequencies that sounds are composed of. So you need quite a bit of computing processing to happen on board the tag. So that's where you have a lot of competing pressures, because we want to as much as we can minimize the size of the tag, but the battery is a limitation. And if you're trying to impose some computing on board the tag, it can be done, but it consumes a lot of battery. So we're trying to optimize all those different competing pressures and trying to extract the critical aspects of the signal. So probably having one of these long duration tags that will have some capability to report some kind of acoustic event is a few years down the line. And actually, to be quite honest, our primary interest right now is in designing new tags. That's what we're working on right now, is in designing new tags that are less invasive, more robust, and less have less of an impact on the animals, any potential impacts on the animals. So that's our primary goal these days. And then after that, if we can incorporate new sensors, for sure, that's also on our wish list.
Wow, that's fascinating. Yeah, it's really amazing where technology is taking us, and it's really great that you're able to kind of ride along on the stuff that, you know, our little digital leashes that we carry around, and take that technology and apply it to other really great uses. I think, you know, for those of you that are still on, it is Giving Tuesday. So if anybody feels that wave of generosity, I'm sure Daniel's lab can always use funding. I don't know, I'm speaking out of turn a little, but I think whale research is expensive, and a lot of students you know, struggling and trying to make ends meet. It's probably funded, but I think they could always do extra. ACSLA, as well as the Cabrillo Marine Aquarium, also benefit from any generosities that any of you have to offer on this Giving Tuesday. So that's a nice way to end. And I'd also like to take a second to thank and introduce, if you guys want to pop on, Kathy and Reed from Cabrillo Whale Watch, who have been helping us behind the scenes, monitoring the chat, doing all the spotlighting. It does take a team. Louisa, of course, you're welcome to come back on if you'd like and say hello and goodbye. And we wish everybody the best. Of course, when I talk about seeing you or having good holidays, I'm not talking about our local Cabrillo Whale Watch volunteers, because we'll see you next week. But the rest of you that are here for our deeper dive webinars, we will see you again in January. So stay tuned. Thank you all. And thanks again, Daniel. This was really a pleasure hearing your stories, hearing all of your
pleasure was all mine. Thank you so much for your invitation.
Yes. Thank you. Happy holidays, everyone.